All right, so welcome everybody who's joined the Zoom. Today, we are very fortunate to have Dr. Michael Snyder as our seminar speaker. Dr. Snyder is currently the chair of the genetics department and director of the Center for Genomic and Personalized Medicine at Stanford University. These are positions that he has held since joining Stanford in 2009, after a highly successful career at Yale as an associate professor, department chair, and director for the Center of Genomics and Proteomics. For those of you who are not familiar with Dr. Snyder's work, he is a pioneer and a leader in the field of functional genomics, multi-omics, and personalized medicine. He is responsible for developing many technologies that many of us use and that are critical to these approaches, including RNA-seq, ChIP-seq, Heritability sequencing, de novo sequencing, to name just a few. He's also a major participant in the ENCODE project that seeks to catalog all of the functional elements in the human genome. So Dr. Snyder's contributions to science are vast. To touch on just a few of these major discoveries, the first high-throughput DNA sequencing of an organism, uncovering hidden complexity in the human genome. That includes revealing a much greater level of structural variation, transcription factor binding sites, and transcription of non-coding RNA. And the first longitudinal multi-omics profiling of a human. So reflective of his contributions to science, Dr. Snyder's publication record is, I would say, beyond extraordinary. His H-index is 184. He has over 600 publications and over 77,000 citations since 2016. And he has nearly 30 publications with greater than 1,000 citations each. And as Rob was mentioning earlier, he literally wrote the book on genomics and personalized medicine. So in addition to his stellar research record, Dr. Snyder has served on a number of advisory committees and editorial boards, including the Human Proteome Organization and Genes in Development. He is also the co-founder of a very impressive number of companies involved in longitudinal monitoring and assessing disease risks, like QBio, we just discussed earlier. And some of these companies, like the protein array company Protometrics and the monoclonal antibody selection company Affomix, have now been bought by Invitrogen and Illumina, respectively. And as the graduate students pointed out yesterday, Dr. Snyder has also been a highly successful mentor. That includes over 70 graduate students, 180 postdocs, 120 undergraduates, and 65 high school students. And he has also been the PI of an NIH training grant in genomics and proteomics since 2004, first at Yale and now at Stanford. So Dr. Snyder, we are honored to have you with us today, and it is my pleasure to give you a warm virtual welcome to the University of Tennessee Health Science Center and the Genetics, Genomics, and Informatics Seminar Series. So welcome.
Great. Well, thanks so much for having me here. That's probably one of the nicest introductions I've ever had. So gosh, feeling a little embarrassed here. Let's see. So anyway, I'm going to share a screen and oops, there we go. Load this up. Great. All right. So hopefully all's good there. Push
Looks great.
Face out of the way. Good. Okay. So yeah, I am going to tell you about our work managing health and of course, I can't resist putting some stuff in on COVID-19 using big data, which is really our shtick, if you will. And I'm going to start with a premise that may offend a number of you, but hopefully you'll partly agree with it, which is I think our medical care system is broken in many, many different ways. And it's just, we talked about this earlier with the grad students. This is just part of this. We're very focused on treating people when they're ill. As such, it's very reactive. When people are healthy, we measure very few things, and I would argue some of these are probably not terribly useful. When you're healthy, you rarely get measured, and this is probably one of the biggest problems. Nearly every decision about your health is based on population-based measurements. Instead, I think we'd all agree it's better to be focused on keeping people healthy, as such, be very proactive. We're certainly capable of measuring many more things than we currently do. And the frequency, I would argue, should depend on what you're at risk for and what your personal trajectories look like. And lastly, and most importantly, I think we should be much more focused on individual-based medicine rather than population-based medicine. And most physicians will say, well, we do that already, but I would argue that we don't, not at the level at which we could be doing things. And so we should be very focused on precision health rather than precision medicine. So just to illustrate this point, this is actually something we think we all know. You've been told since you were little, your oral temperature when you put a thermometer in your mouth is 98.6. But if you actually look at the data that are out there, this is a very typical study. The number is more like 97.5. I personally am 97.3. But more importantly, there's a spread. 94.6 is 25th quartile, 99.1 for the 75th quartile. And so what that means in today's world, if this is your normal baseline temperature, you go to a physician and they measure you at 98.6, they'll tell you you're healthy, everything's great. What are you doing here? Go home. But you're at 4 degrees Fahrenheit over your baseline. I guarantee you're probably not healthy. And so this is a theme I'll hit many times. It's really important to know what your baseline is so you can detect shifts from that. So our health, I think you all know, it's influenced by many things, your DNA, but all these other things as well. Exposure is very relevant to today's world with the pathogens. Stress, food, lifestyle, like exercise, these all influence your health. But we're in a world now where it's possible to quantify a lot of this, and we're actually working on some of this. Some of it's easy, like activity you can measure with various devices. Food's pretty clunky still. There's indirect measures for these other things. We're actually working pretty hard on this. I won't talk about that today. But even if it's hard to measure all of these things, you can measure their impact by actually doing detailed profiles on people. And so a number of years ago, it actually started 11 and a half years, but for most of the group I'll tell you about today, about 8.5 years ago, we launched this, what we call personal omics profiling, where the idea is that in addition to your genome, we'll make many, many other omics measurements as well. And these are generally out of blood components. So epigenomes, DNA methylome, and transcriptome will measure PBMCs, peripheral blood monocyte cells. Proteome out of those cells, and cytokines and metabolites and lipids will also measure out of plasma, or sometimes serum. I guess these are all plasma. We'll measure the microbiome, gut, nasal, and more recently we've added tongue and skin. It's turned out to be quite interesting. We do this on top of deep clinical tests and questionnaires. And we have some advanced tests a number of participants do, like stress echocardiograms. They all do an OGTT, as you'll hear in a minute, oral glucose tolerance test. And about 8 years ago, we got very involved with wearables and biosensors, and I'll spend some time on that as well. So one aspect of this is deep data. We collect this, as I say, on, it turns out a smallish cohort. It's meant to be a pilot study, although we learned a ton, as you'll see in a minute. And the other aspect is longitudinal. We're collecting these samples every 3 months while people are healthy. And then if an adverse event comes along, like a viral infection or other things do happen, we'll take usually 5 to 7 additional samplings. So you might say, and again, we've been running the study for about 8.5 years, a little bit more for some, a little bit less, but roughly that number. And you might say, Mike, why are you doing all this? Well, the answer is we're trying to understand, what does it mean to be healthy? How does that change over time? How does that differ between different people? What happens at the earliest times of disease, like viral infection? And perhaps most importantly, at least relevant to many medical schools, is, can you use these advanced technologies to be able to better manage people's health? Like, is genome sequencing really valuable? And if so, how often? So with regards to this last point, I'll jump right to the chase. Just from the first little over 3 years of data, we had 49 major health discoveries. So, and it covered a wide range of areas. So heme-oncology, cardiovascular disease, metabolic disease, so on and so forth. And some of these turned out to be a pretty big deal. So for example, we caught some with early lymphoma, others with, these are precancers, MGUS it's called, and smoldering myeloma. They can convert to aggressive cancers. 2 people with serious heart issues, one from genome sequencing, another from wearables. And that was one of the points we learned from this. No one technology found all these things. It did take the combination of different technologies to actually reveal the full spectrum of what was going on. Another important concept I should mention is that this is all done, these are all found pre-symptomatically, meaning people, these things are picked up by doing these deep profiles. We didn't wait for them to get symptoms. And so they were caught early in most cases, and we think that's obviously quite valuable. So it turns out, as I say, all technologies were powerful. Genome sequencing was one. So the first 70 people, 12 individuals learned something pretty important about their genome from just as what we call the Mendelian disease, where you have single mutations that lead to highly penetrant. The most famous, I'm sure you're all aware, is BRCA. An individual in our cohort had no idea, but they have a BRCA mutation. They were adopted, so they didn't really know what their parents' situation was in terms of familial breast cancer. And then there were other cases as well. These are what are called the American College of Medical Genetics genes, for those of you who follow that sort of thing. These others are just other disease associations. We go very, very deep on people's genomes for our interpretation. Now, 3 individuals, it turned out what they learned was a big deal. So one individual turned out to have a mutation in this cardiomyopathy gene, and it turns out his father had died of a heart attack in his 60s, and his aunt had one in her 50s. And so this person then did a follow-up test, and sure enough, they actually have a heart problem, and they're on drugs now as a consequence. So the genome sequencing did reveal something very, very important. This individual turned out to have a mutation in this gene, which puts them at high risk for certain cancers. They did a whole body MRI follow-up, and it turns out they had early thyroid cancer. And so they had that removed. They're able to keep the rest of their thyroid. And of course, then they don't need replacement therapy. So that's a good thing there. There were 9 individuals coming into the study that were type 2 diabetic, or at least called type 2 diabetic. One of the individuals turns out has a mutation in this gene, HNF1 alpha, which puts them at very high risk for, well, diabetes, but it's a different kind of, it's actually called MODY. It's not type 2. And so this person's been on the suboptimal medication for years and is now switched over because they have better information. So genome sequencing was powerful. As a 13th individual learned something very, very important pharmacogenomically as well. So so that's the Mendelian disease. We've come up with new ways for looking at complex disease. So you probably know most disease is actually complex disease, things like diabetes, coronary artery disease, all these things. There can be Mendelian forms, but the most common form is, in fact, what's called polygenic. That's thought that these are due to lots of variants of low effect. And so what people have been doing, this is now what the GWAS folks do, they've developed these polygenic risk scores. Many of you may know this, where you sum over, now these days, you sum over millions of variants or thousands to actually make predictions about people's risk. And it works for the top end of the distribution. It doesn't work that great for the middle end and stuff, but it works if you're at the very, it turns out, worked for me. I'm at the top end distribution for type 2 diabetes, and you'll see in a minute, I'm type 2 diabetic. So it works for us. Now, there's 2 problems with this approach. Problem number 1 is it assumes genetics is linear. It sums over these things. And we know from all the model organisms and other studies, genetics is not linear. If you have 2 mutations, same gene, 1 + 1 is 1, if they're in the same pathway, I should say. And if you have 2 mutations and things that are different, they can sometimes be synergetic. 1 + 1 could be 15 in terms of effect size. So that doesn't take this into account. It also doesn't take into account the rare variants I mentioned earlier. So we've come up with new ways to analyze genomes that involve machine learning, it turns out. And I'll just show you the first one we published, but you look on bioRxiv. We have 2 others out there that are pretty cool. This is one we went after. It's called Abdominal Aortic Aneurysm, because Phil Sal had collected material on these patients. And it turns out it's a big deal. And it's a big deal because this disease it's semi-prevalent. 10% of the elderly 65 or older will get it. And the reason it's a big deal, it's usually discovered when people's aorta bursts and they die. So nearly everybody dies when they discover this. And obviously that's a terrible way to discover you have this problem. So we wanted to see if genome sequencing could be valuable here. So we did something that would scare all the GWAS folks. We basically decided to try and look for rare variants. We did do some GWAS, but our cohort was small. We had 268 cases, 133 controls, 401 people total. And we sequenced them, to 50x coverage. And we did look for association. I knew we wouldn't find anything because we're underpowered for these common effect variants. But what we did look for was, in fact, rare variants inside the coding region. So even though we did whole genome sequence, we focused on the exomes. And what we did was we took the pathogenic variants there, along with a little bit of EHR data. And we basically say, give us all the genes. I forgot to say there's known to be heritable. It's thought to be 60% heritable. And yet the genetic underpinnings have not been well described, that people have slugged at it, but haven't really come out. So we did this machine learning approach where we basically took the pathogenic mutations together with these other features. And we said, give us all the genes in an agnostic fashion. We use what's called tenfold crossover method for the machine learning. And wouldn't you know, up popped 60 genes that were highly enriched in the cases relative to controls. And so you can never get this out of a simple GWAS kind of association. So again, 60 genes were highly enriched, like 10 to the minus 13. When you look at the ensemble. If we look at the kinds of genes involved, it turns out they're exactly what you would expect. They're in networks that involve blood circulation, blood pressure, cardiomyopathy, or aneurysm. So they're in exactly the same pathways. Mouse models of these 60 genes give aneurysm kinds of defects, or they give, I should say, and it turns out both in humans and in the mouse models, the 60 genes we identified, they're all misexpressed, or not all, but highly enriched for misexpression. So again, it's exactly the sort of thing you would expect. And then we went on to set up predictive models where we, just from a genome sequence, which is a red, but even better if you add in a little EHR data, you can basically get a pretty decent predictive score for someone's likelihood for getting AAA abdominal aortic aneurysm. And this is about as good, I'm not sure if I didn't show that slide, but it's about as good as, say, the Framingham heart kinds of tests that exist now. So we think that this is potentially usable. We've gone on to do a slightly different version in terms of common variants. So using GWAS data for something called ALS, Lou Gehrig's disease, and there we discovered 690 genes, up from 13. So it really pulls out a lot of genes you'll never get, this machine learning approach. And we've done it for severe COVID, not for the elderly, but for, as you know, a lot of young folks wind up getting hospitalized. And when you look at those, it turns out we can get the genetic underpinnings there, about 1,300 genes, up from something like 7 genes again. So this approach of machine learning for analyzing complex disease, we think it's going to be very, very powerful. You can take a look on bioRxiv for the 2 papers I just mentioned. So as I said, genome sequencing was quite powerful, but it's really only part of the equation. So 13 people out of 49 learned something important. Imaging, in this case, it was imaging plus some biomarkers, protein markers that were elevated that flag this case as a case of a large spleen, and turns out they actually had early lymphoma. 2 people had precancers, as I mentioned, MGUS and smoldering myeloma. This one individual had given 2 samples at the time that we analyzed, had way too much IgM. That was just so separated from the other, essentially 998 folks. So his samples were way out there. Also a young person. So these are people, who were thinking they certainly have and still can, of course, have a big future ahead of themselves. But they do have these changes, and this is useful to know because you get screened more if you have this, because it can convert to aggressive form in 15% of cases. So this is a case, remember I was kind of poo-pooing outlier analysis. We do find things from outlier analysis that's important too. Imaging revealed 6 people with plaques in their arteries. I'm one of the 6. This isn't me, but my plaques kind of look like this, basically. So you increase statins, and in my case, it seemed to work, and they go away. We're very interested in type 2 diabetes because, as I mentioned, I myself am type 2 diabetic. And so coming into the study, 9 people I mentioned thought they were diabetic, but 2 were diabetic and didn't know. So the number is actually higher when they entered the study. And then a whole bunch of people are pre-diabetic. That's what this means. And it turns out 9 out of 10 pre-diabetics don't know. I'm pretty sure a lot of you on this call maybe pre-diabetic, not a lot, but some of you on this call most likely will be pre-diabetic, have no idea. This is a big deal because you can convert from pre-diabetic, 70% of people are pre-diabetic will become diabetic in their lifetime. So we're also very interested in how do people become type 2 diabetic? Do they gradually get there? Do they spike them? What's going on? And so it turns out over this essentially 3+ years, 9 other individuals were clinically diagnosed as diabetic, and others had diabetic range values, and some others got pre-diabetic as well. And if you look at what's going on, well, it turns out of the 9 that were clinically diagnosed, 7 just gradually got there. Their orange is their fasting glucose. Here's hemoglobin A1c, a marker for glycolated hemoglobin. And so, yeah. Sorry, sneeze coming. Not COVID, I don't think. I think it's allergies. I do get tested regularly. So anyway, I'm here anyway, so you're all safe.
Fortunately, we're pretty safe.
Yeah, I think so. It doesn't go through the electron. So all right, or go with the electron. So here we go.
It's 5G. I'm pretty sure it goes through 5G. So I'm not sure we're safe. Sorry, go ahead. There's a bad joke.
Okay, got it. All right. So anyway, yeah. So 7 people just gradually got there. Some of these then went on metformin and got it under control. And 2 of them got there. 2 of the 7 actually gained weight. So that's how, and their microbiome diversity dropped it and so on. So they kind of got there classically. But 2 others spiked their way there. This is me. I'm one of the 2. That's 8 years of data. And I first became diabetic after very nasty viral infection, actually. So and viral infections have been associated with type 1, not type 2. So this is kind of new at the time. And I know this looks like a little blip. It's about 10 months of data there. And bottom line is I had to change my lifestyle. Excuse me. Yeah, basically I cut out all sugars. I had been biking, doubled my biking, and started running. Gradually, that's not a few days, that's like 10 months to a year, brought it down to normal. And in fact, it was so boringly low, I stopped looking at it. And then once you know, someone else who looked at it here said, Mike, do you realize you're back to diabetic again? Because my hemoglobin A1c, it's arbitrary, but 6.5 is classified as diabetic. So first time I was diagnosed. And then it came back as I say, and we looked back at when it shot up. So it's, first of all, it's pretty clear something's spiking mine, and the hypothesis is I'm genetically predisposed. In this case, with the viral infection is what triggered it. Over here, I had a second viral infection, but I also stopped running. So it could be either or a combination of both. And so I started running again, and I brought it down, but never been down to baseline. So there is a running effect here. And then basically I was kind of running 4 times a week, 45 minutes a time, 45 miles a shot. And bottom line is, my glucose kept creeping up anyway. So about 5 years ago, a little over 5 years ago, I switched from running to actually weightlifting because I heard muscle mass was better for glucose homeostasis. So I shifted, and that actually failed miserably. It kept creeping up. So I finally bit the bullet somewhere in around here. I mean, it was here. And I took a metformin, which is the frontline drug for type 2 diabetes. And guess what? I'm a non-responder. So it actually crept all the way up to 7.5. And then finally, how did I solve my problem? Well, more data. I'm a big believer in data. So what we discovered is that I am insulin sensitive for the most part, and I actually make insulin fine, but I don't release it from my pancreas, which is not common, but it's not, totally rare either. And so my glucose disposition is quite poor, quite delayed. And the bottom line is that, again, I don't release it from my pancreas. And there's a drug for that. It's called Rapinolide. And it actually works pretty good for me. And so this is what we call precision diabetes, but better understanding exactly what's wrong with you, you can treat people appropriately. And I also now respond well to SGLT2 inhibitors. So both those work pretty well. And so that's kind of, again, precision diabetes for better managers. And by the way, you don't have to be, this stuff's not super expensive. The way we do it's expensive, but to figure out if you're insulin resistant or not releasing things from your pancreas is a very simple test, actually. So my own view is we should be doing this for everyone. All right. Now, one of the things we learned by all this profiling is that people are very personal in their profiles. And so no matter which you use, we all have personal profiles and they're quite stable. So this is just showing 12 people, at least 10 healthy visits. Every dot's a different visit, every color is a different person. And I'm the light blue. There's 6 years of data for me. You can see it doesn't matter what cytokines, clinical lab, what have you. My dots all cluster together. Even if I were to show you this in third dimension, these things cluster by person. okay, not by visit or what have you, and they're pretty stable over time. And so that has interesting privacy implications, because if we were to swipe your chair where you're sitting now, we could tell it was sat there if we had the right reference. Not only that, it turns out that if you undergo a perturbation, say a viral infection, or in this case, we had people run to their VO2 max. They did an acute exercise bout, and half their molecules change. But if you cluster them, it turns out they still cluster by person, not by whether they're at their VO2 max or what have you. Same with the viral infection. They still tend to cluster by person. So this is a big deal, because what it means is that your profiles are very stable, and the difference between you and a perturb, you say a viral infection or some other thing, is actually less than the difference between you and other people. And so what this means is the reason GWAS folks need thousands and thousands of cases, because if you want to tell the difference between different people, that'll be bigger than the disease. The difference between different people is bigger than the disease versus healthy. And so it's very easy to spot your disease state when you compare to your healthy baseline, but it's hard to compare if you compare your disease state to 500 other healthy people, because again, the differences between people is so large it masks the effect. So I would argue you get your healthy baseline now. And that's a hard concept for people to appreciate, that you really comparing with your healthy baseline is just by far the best thing you can do. So I told you these profiles are pretty stable, but there are two times they change. They'll change seasonally, and they'll change with age, with time. And so one fun thing we did was we said, all right, we have all these deep profiles on people over multiple years. Can we actually ask a very naive question? How many biological seasons are there? And these folks are all collected in Northern California. So if you think about it, we've been told since we were little, there's 4 biological signals: winter, spring, summer, fall, and they're 3 months each. And if you think about it, this is like totally arbitrary. Maybe there's 15 seasons, who says? Maybe 15 seasons, 3 seasons, what have you. And why on earth would they be 3 months each? That doesn't even make any sense, right? That's just totally arbitrary. So we could actually go at this systematically and just say, well, how many seasonal patterns do we see in the data? So we first, here's all the samples, all these dots. I'm this one here with all the, I've given several hundred samples by now. And so what we've done is basically we look for all the molecules out of many, many thousands, tens of thousands of molecules of microbes. We see which ones have seasonal patterns, and there's 1,192. And you can see here, they go up. A few of them were known already, although most of them not the resolution at which we measure them. And most were completely brand new. This is all new. All right. So we said, all right, we have these 1,192 molecules of microbes. How many patterns do they fall into? And the answer is 2. And you can show this mathematically using these elbow plots, that there are 2 major patterns in the data. And there's some other molecules that fall outside that. But bottom line is, and you'd say, well, of course, it makes sense. It's Northern California. There should be 1 in winter and 1 in summer. And there you would be half right. There is a winter pattern. So these are molecules that peak at the end of the year, November, December, into January, and then come back down. And it's exactly the sort of stuff you would expect. So a lot of it's viral infection associated. These things here, acne is known, believe it or not, to peak in winter. And these other, and then you say, well, what about the other pattern? Well, it turns out that's late April, early May. And that's when allergies and asthma are prevalent. And that makes a lot of sense too. Pollen's out there and all these things bounce up. But there's other markers there too that are quite interesting. Cardiovascular disease, metabolic disease, type 2 diabetes, all markers peak around this time, as do seizures and sleep. And I don't understand fully the last 2, but these kind of make sense in hindsight, where you can basically say, well, even in California, people aren't that active in winter. It gets cold for us here, like, 50 degrees. And not during the day, it's still usually in 60s. But anyway, it gets cold at night. And not only that, it rains. And so people, we think, don't exercise as much in the winter. And so these not so great molecules will build up. And then spring comes, people get more active, they go back down. So we think this is important because there's a lot of important information, like there are clinical markers associated with these patterns. And so we think you should take this into account in your thinking when you're trying to interpret people's health. That, yeah, all right, they're getting high, in spring, or if they're at risk, well, don't let yours get so high. Go work out the gym some more. But you can incorporate this into your thinking for the healthcare. All right, the other time when things change, we found 600 markers that will change over time, and we're referring to these as aging markers. And our study is unique because we're measuring the same person over time, so we can see how people age at an individual level. And so this is person one, person two. Person one is me. This is 4 years of data in this particular case. And I'm a pretty typical age. My coagulation pathway, metabolic, what have you, all go up with time. But it turns out that, if you look at someone else, well, they're differently. This person's top pathway is cardiac hypertrophic signaling pathway. So they're a cardio ager. And so what we discovered is everybody's aging differently. There's personal aging going on. And just like the seasons, we can say, well, how many patterns do they fall into? And the answer is we found 4 major patterns. Now, I know there are more than that. We only have 43 people with enough data. By the way, we think you only need 5 time points over 2 years. We'll know how you're aging. And so what we discovered is that there are 4 major we call them ageotypes, 4 aging patterns. And I know, once again, I know there are more than 4, like the cardio, but we only had one example of that. And so the 4 are kidney, liver, metabolic, and immune. And the net result was each column's a different person, each row is a different ageotype. And so the 4 people over here, they're aging in all 4 categories. This one's aging in 3 of the 4. I'm somewhere in the middle. I'm not much of an immune ager, but I think it might be that one. I age a little more in the other 3. This is a kidney ager, kidney and metabolic, and so on. So again, we can classify people into different patterns. Not only that, there's clinical information associated with these patterns. So again, we try and anchor things into things people know how to act upon. And here's hemoglobin A1c. This is the easiest to interpret. The group as a whole goes up over time, and that's known already. The thing that the groups will go up with hemoglobin A1c. It hadn't been shown as well at the individual level. And when you look at the individuals, there were 4 people, dark bar means they're statistically significant, 4 people that actually did go up, and then 4 others that decreased over time. And if you look at what happened to these 4 that actually reduced their hemoglobin A1c, it totally makes sense. 2 went on a diet, 2 lost weight, 1 person didn't really start exercising, and so they've actually improved theirs. Here's one that doesn't make as much sense, called creatinine, which is a marker of kidney function. And it turns out the group as a whole goes up for this marker. But if you look at the individuals, 10 of them actually go down. So again, personally, you may not be behaving as a population as a whole. When we looked at the 10, 8 of them are on statins. So we tend to think maybe statins is having some role in improving kidney function. That's the hypothesis, needs follow-up tests as well. We also discovered that people are aging differently. So this is an aging marker system set up by Morgan Levine. She calls it PhenoAge. And this is people's chronological age. This is their biological age. Their biological age versus, again, the year they were born, if you will. And what we discovered is that, some people are biologically older than their chronological age. That's not a good thing. Kind of want to be on this sideline. These folks are actually younger. So the net result is we can actually, again, different people are aging differently by these marker systems, and we can follow that. Some people say, Mike, how are you going to scale this? Is really expensive. You're right. It's a research project. And but the way we scaled them in Silicon Valley is you form a company and so you can get out to a bigger group. And so we formed a company called QBio, and they're basically doing a medical version of IPOP, the same profiling. So it's more medically oriented, and they added whole body MRI. And just in the first 100+ people, they made some pretty big discoveries. You can see them summarized here. And usually it's multiple lines of evidence, like imaging plus other markers, really tip you off that some of these things were happening. In other cases, the longitudinal information was very, very important, like this pancreatic cancer was picked up because one could see this growing nodule, if you will. So this was, in fact, a big deal. And so we think this is how it could be scaled by running a version of this. As I say, it's not cheap, $3,500, but nonetheless, it's better than dying, I guess. So this is all picked up pre-symptomatic. Now, the last area I want to tell you about, oh yeah, we're in good shape, is our wearables. So we're doing a ton of work in the wearable space. And we got involved in this about 7 or 8 years ago when the Apple Watch wasn't out yet, but there were Fitbits out there. Most people are using it as fitness trackers. They wear them, figure out what's going on, and then, toss it in a drawer after 3 months. And we said, well, these are probably powerful physiological measurement devices. They make hundreds of thousands of measurements a day. Some of them make as much as 2.5 million measurements every day. The smartwatches do. They're worn by lots of different people already. 50 million people in the US have a smartwatch they're actively wearing. They're capable of tracking many things: heart rate variability, respiration, blood oxygen, skin temperature, so on and so forth. So we can profile a lot of different things. And some of these are pretty accurate: heart rate variability. Some of them not so accurate, but the deltas are still pretty good. Skin temperature you can get pretty good. But these two, they're not accurate, but you can follow the changes pretty well. And so you can track many, many different things. And so we discovered early on that you can actually tell when you have an infection from a smartwatch. And in fact, this is how I figured it out, from Lyme disease of all things. And so the backstory here is helping my brother put up fences in rural Massachusetts. And 2 weeks later was flying to Norway through Frankfurt. And you may or may not know, but on aircraft, your blood oxygen drops because they reduce the pressure in the cabin. And on this kind of aircraft, it would normally drop to that. But it turns out that, again, from wearing a pulse ox and a smartwatch, we've discovered that my blood oxygen dropped abnormally low on that flight and never came back to normal after I landed. And I also saw my heart rate was running high at the time, and later learned my skin temperature was elevated as well. So this is all pre-symptomatically. And then later, I did get a low-grade infection. So I went to a doctor in Norway. I warned it might be Lyme because, I had been putting up these fences in rural Massachusetts where 55% of ticks were Lyme infested. And he drew blood, said my monocytes are up. Yep, you've got a bacterial infection. You should take, he wanted to give me penicillin. I said, no, I need doxycycline. Got a little tense, as you might imagine. Doctors don't like their physicians telling them what they should be doing, but he did give in. And you take it for 2 weeks, and I did. And when I got back, I actually tested, and sure enough, I was positive for antibodies, and even some antigen was left there. And it's well controlled because a few days before I left, I'd given blood, and we tested that, and it was negative. So I clearly seroconverted during this time. So this is kind of cool. We picked up my Lyme disease from a simple smartwatch and a pulse ox. So we went on to look at 2 years of data at the time. How often does this happen when you're ill? And it turns out I was ill 4 times. I had 73 measurements over those 2 years. And this is C-reactive protein. I was ill 4 times. This is a viral infection. That's a viral infection. This is the Lyme case. And there's a 4th time where it's asymptomatic, if you will. And so what we discovered is that every single time I was ill, I had high outlying resting heart rate or a skin temperature. So again, you can't miss the Lyme ones are especially powerful. They really shift you. And so we can pick this up. And so we went on to write an algorithm called change of heart. We tried it for skin temperature, but it doesn't work as well in skin temperature because I think not everyone holds their watch as tight as they should, or gets their watch strapped as tight as it should. So anyway, but for heart rate, it worked really, really well. And this is a sliding window delta plot, if you will. So you can see that you're looking for this change from your previous measurements. And turns out this is symptom data in red. Every single time I got ill, I could pick it up with my smartwatch. And so it worked for me. This is all retrospectively on 4 cases, even the asymptomatic case. And then we had 3 other people, one of whom got sick twice, worked every time for them. So as you might imagine, then the pandemic came back in March of last year, and we had been improving these algorithms, building a system to try and set up this kind of monitoring. And then when the pandemic hit, we went at it full force, really did a lot here. And, here's where we were then, even now, and it's less than that now. I know we're a little day, but I think we're still around 100,000 measurements a day. You can look it up. All right. So that's still a lot. And of course, people are going to worry. It's going to be around at some level, probably permanently. So if you think about what we're doing now for measuring when people are getting ill, the gold standard is PCR. But for us now, we at Stanford with all the students back, thinking about 3 days to get your results back. And that's really slow. And so thermometers you can use too. And you know, if you think about it's 300-year-old technology, the problem with these kinds of thermometers, they shine them on you if it's cold outside. I always register very cold. So I'm typically registered about 33 degrees centigrade. And, you know, that clearly can't be right. Be probably close to dead. But the point out of all this is that I think these things are probably not terribly accurate, yet that's what people are using. So when we think smartwatches could be a way out of this, and that's, or not, at least something that can help with the diagnostics. And so that's what I'm going to tell you about now. So we very quickly launched, when the pandemic hit, we had a study running, we launched it to make it broader. We partnered with leading companies, Fitbit, Garmin, what have you. And right away, we enrolled 5,300 people, 32 of which had perfect, what we call golden data sets. These are folks wearing a Fitbit. We were working with Fitbit at the time. These are people 32 people wearing a Fitbit at the time they got COVID, and they have both a diagnosis date and a symptom date. And so what we're trying to do is see if we can detect COVID using these folks. And so this is our very first case. You can't miss this. This is resting heart rate. Here's day 48 when they're diagnosed ill. Here is day 47 when their symptoms first appeared. But you can see their heart rate jumped up 9.5 days ahead of time. You cannot miss that signal. So this person's been presumably running around, basically spreading the disease because they didn't know that they were basically they were asymptomatic with COVID. Didn't know. And I'll show you in a minute, we have a real-time alerting system where we build a baseline and look for a jump up over that baseline. In this case, hour by hour basis. All right, so of the first 32, 26 of them, we could pick up with a smartwatch. So that's pretty good, about 80%. It turns out we picked them up a median of 4 days prior to symptom onset and 7 days prior to diagnosis. And it's not specific for COVID. Other illnesses can trigger this as well. Yeah, so it's a general, and it also show up with stress tests and things like this. So the bottom line is we could pick up this we could show we could detect COVID with a smartwatch. So we did go on to write this real-time detection algorithm. This is the first version of something we wrote. Here's a case where diagnosis day in purple, symptom red, heart rate jumps up about, again, I think this one might be 9 days. This one's 7 days. There are other alerts that go off. And we know that alcohol, travel, excessive alcohol, not a few drinks, but if you really tie one on, that'll trigger these alerts. So yeah, hiking the mountains can do it. And psychological stress will trigger these alarms as well. So we also noticed that quite a few people have holiday mumps, as we call them. That could be because of stress from the in-laws, alcohol, travel, what have you. So that certainly fits. Here's a case of, another illness, not COVID, and it works for that too. Not quite as advanced as COVID because, because we think, again, other illnesses they will show this, but they don't have as long pre-symptomatic periods. So they pop up faster. And you can see there's post-infection stuff too. Here's a healthy person has a peak. Again, maybe it's stress, not really clear. All right. So we've gone on to make now yet another real-time detection algorithm that's lightweight, because what we're doing, we build an infrastructure now that can scale to the entire planet. Basically, certainly do millions. I'm guessing it should be able to do billions. So we want you to sign up for a study, innovations.stanford.edu/wearables, because then you can, join this. And the way we've set this up is alerts will go off. You actually have to test and sync up. I forgot to do mine today, but you sync up with the algorithm we've said. It's all app-based on the phone. And if you have so we're building a personal baseline. Some of the algorithms are hour by hour, some measure over an entire night. And we're looking for a jump up over your baseline from either the previous 28 days of measurement or 7, depends on the particular algorithm, and see if we can detect these things. And what we found is that 80% of the cases, 67 of 84, we can see that we can send off an alert at or before the time of symptom onset. So here's one example. Here we time shift dates, so they're not real for PHI issues. So here's a person who got a red alert here, was diagnosed here, and the symptoms came here. So it really does seem to work. It even picks up asymptomatic cases. 14 of 18 asymptomatic cases are associated with these alerts. Here's one example. I forgot to say we have the algorithms working for both Fitbit and with Apple Watch. And I'm sure it'll work with some of the others. We just don't have as much data like Garmin and such. So again, you can see this. Here's a case of asymptomatic case that we picked up. Let's see, here's another one. Oh, there's a diagnosis day. Here's their day. And so again, we think we can pick up asymptomatic cases because your heart rate jumps up when you get ill. We published a first study on this over a year ago. And others have come out since with Apple Watch and Oura Rings and things like this. So we think this is going to be a general paradigm, again, being shown by a number of groups. Now, we think smartwatches and infectious disease is just the tip of the iceberg. There's a lot of other stuff you can learn. And here's an example here. We did some machine learning to see if we could correlate features from a smartwatch with, in fact, these clinical measurements. And what we discovered is that these measures, which are measures of anemia, hemoglobin, red blood cell, hemocrits, that's your so we can actually make predictive models on these from smartwatch data. It's not enough to make a clinical diagnosis, but it's enough to see, hey, something's up. You should go get tests in a clinic. And same is true for hemoglobin A1c and fasting glucose. We can get some association just from a simple smartwatch, which is what's going on. The last area I'll tell you about is continuous glucose monitoring. I think we're okay time-wise. Because I saw there's time for questions after, but this will wrap in about 5 minutes. So I mentioned before, we have a lot of interest in type 2 diabetes, and there are these new devices out there called continuous glucose monitors. By the way, I'm totally keen on this stuff. I've seen my 4 smartwatches here if you're looking on the screen, and I wear a CGM normally. I'm in between right now. I use 8 of these devices every day. But continuous glucose monitoring is very powerful because they measure your glucose levels every 5 minutes. So we went out and put these on normal people, pre-diabetics, a couple of diabetics. And what we discovered is, some people have nice stable baselines, good glucose control. Some people are moderate spikers and some are pretty severe. So we set up, we developed a method, an algorithm method for classifying people into glucotypes, whether low glucose levels, if you will, moderate or high. So we've classified people in these different groups based on the information. The other thing we've done is we've given people different foods, and what we've discovered is that different people do spike to different foods. Similar work has been found by Aaron Siegel, did a beautiful study on this. So bread and peanut butter will spike some people, protein bars, other people, grapes, some pasta, others. So it really varies from person to person. Your microbiome is part of it, but it's not the whole story. So there's other stuff out there. Nearly 80% of people spike to cornflakes and milk. So we think this stuff's like poison for you, probably should be banned. Anyway, so that's, well, it is what it is. So one of the things we discovered is that people who are normal by fasting glucose, hemoglobin A1c, even oral glucose tolerance tests, and that surprised me, they can be just as severe spikers as diabetics. And so, here are 2 cases. These people are classified as non-diabetic, but it turns out they're very, very severe spikers. I suspect there's clearly strong glucose dysregulation going on them. So lastly, what we've done is we pulled together, we built a dashboard, again, that's scalable to millions of people. We have, something like 7,000, we want millions, want all you guys to sign up, get your brothers and sisters and family, friends all to sign up for our wearables study. And what it does is the dashboard will bring in your wearable data, your clinical data, microbiome, all into a common screen here for you to look at, and then you can play with, make your own correlations. We think this is powerful because you can share this information with physicians. So things you're collecting now, a lot of people say, Mike, these devices aren't that accurate. And I said, well, they're more accurate actually, at least heart rate is at home than it is in a physician's office. And so by sharing this kind of information, you can better, assist them. So this is Mike Snyder's future. I envision a future where people get their genome sequenced before birth, and then together with other deep profiles, molecular wearable data, we'll be able to better, predict disease risk, diagnose disease early, monitor and treat disease. So we think big data can really transform the future. So I've been fortunate to have some amazing people in my lab. These are folks who led the iPop project, Wenyu, Brian, Kevin, and Tejas, and others. And George Weinstock has been a fantastic collaborator, as has Trace McLaughlin, who also worked with John Bernstein. And yeah, I guess that's that. The Wearables team is here. We, as I say, when the pandemic hit, we just really quickly scaled up, hired a whole bunch of people. And this is, it's a little data, meaning some of these folks aren't here and others here, but for the most part, it's right. So you can see there's a lot of key players over here. But also, again, go to innovations.stanford.edu/wearables and you get right in there and sign up in our study. And so that's the story I wanted to tell you. I believe I've saved enough time for some questions and happy to answer any if you have any. So thanks again for having me.
Thank you. That was fabulous. Questions?
See what's in the chat here.
Oh, yes. Yeah.
All right, Rob, I'm glad you like it.
I have a question. We at St. Jude, right down the street, we had gotten them interested in trying to predict fever in children undergoing chemotherapy. So, frequently they're you're talking about a relatively general healthy population. In kids undergoing chemotherapy, a fever is indicative of a bacterial infection, which could be a life-threatening event. It would be very interesting to see if you could actually drill down in the population of children with undergoing chemotherapy, or even the adult population. They tend to really differ, so you'd want to do it separately. But I'm wondering whether you've ever been approached by cancer researchers with that in mind. We got a lot of interest at St. Jude. It was the study power was daunting because unfortunately well, I guess it's good, but children don't get that overwhelmingly septic anymore. Yeah. But be that as it may, I was wondering if you've ever thought about that.
Yeah, great question, Robert. So it turns out we have a study running with a transplant group who's in exactly the same situation. They're immunosuppressed. They're very vulnerable. They get an infection, then they'll die. So we have a study running with them. It now has 2,000 folks enrolled. And so I totally get it. I'm a big believer in this, and so it's going to work at some level. Again, it's not a perfect system, but it's better than what we do now, which is nothing. So happy to talk with you further about how to set up a study like that. Luckily, we have a template for it already. I can put you, it's a collaboration with Penn and Brendan Keating. So love to make it happen with you. And I'll give you a shameless plug, which is I have a company called Sensomics. The hardest part's getting the data, the raw data. Fitbit, Apple, forget it. They'll give you, you get whatever you get from HealthKit, not that raw at all. Very processed, very low resolution. So that's been a big problem. So, and because of that, we set up our own company to be able to pull this in, set up an API, and the data comes right to us too, doesn't go to the company. So solves all kinds of privacy issues. So, yeah. So I think we're pretty well set up if you want to try and run a collaborative study like that, Robert. Happy to talk further.
Thanks.
I think there's the future. As you might imagine, a lot of groups, not necessarily cancer, but people who have, say, had heart conditions, when they go in the outpatient, they're very interested in monitoring it, right? Those folks would rather be at home than in a hospital. So we're talking about the several major groups around that.
Yeah, I think the high-risk groups, people who are undergoing intensive follow-up and are at high risk, will be the most receptive. And they're also the ones who they'll keep the smartwatches on their wrists and do things like they're supposed to.
Yeah, they should be very motivated. The one minus is that we have to make sure we can get stable baselines on them so we can detect the jump ups, because they can be also a fairly tricky group depending how they're managed. But yep, total 100%, we're totally aligned. Yep. Okay, I see I could go into Rob's on cost. Yeah, so obviously $3,500 is pretty expensive. I would argue it's not that bad. You might have thought it could be a lot worse, especially when you're talking whole body MRI. But it's pretty easy to justify for the high-risk cardiovascular group, because if they get a heart attack, it's super expensive. And we talked about this a little bit before the seminar with grad students that, if you go into intensive care, the bills just skyrocket. So one could argue, keeping people healthy is probably a good thing. What you really want to do is keep people healthy. You probably know the US spends more money on healthcare than anyone else, and yet we don't have the best management because we're dumping it all in the last year of life. So you really want to keep people healthy, and then boom, die. That's how it should go. And that's, we think, the kind of thing could be done with these sorts of technologies. So that's kind of what I'd like to see it do. But we talked about this before, again, the healthcare system's broken. It's not incentivized to keep you healthy. It's really incentivized to treat you. As the head of the hospital tells me, like, nobody pays me until they walk in the door. They don't walk in the door until they're ill. We got to shift that mentality. But then it's super expensive.
And so I have a comment regarding exactly that. So, it was very interesting to see what you presented, at an individual level. I'm not doing any fancy omics, just the regular tracking of simple things. I do it for myself, and I've done exactly the kinds of things that you've done. Now, there are some system level issues. For example, I showed these graphs to my physicians, but they're not interested. They're not interested, they're not engaged. As you point out, they're not incentivized to engage in this manner. So we need some structural changes to make that happen. The second thing is the scalability. So there's two issues with scalability. The one, I think, okay, yes, you can put some of these variables on a mass level. But the second thing is, that I would argue that is not really efficient, efficient way to deliver healthcare. The more interesting things is you're finding like cornflakes and milk is poison. Like if we actually intervene at that level, it is much more efficient than going to every person individually and trying to find out what's wrong with them. I can see that a personalized approach is useful, but there has to be a system level change in order to make the personalized approach more effective. What do you think?
You're raising a good point. I probably have a different perspective on this. I think we should have these minimally smartwatches on everybody. And here's an argument is you don't drive your car around without a dashboard, right? You got to know if the engine light, you got to know if you're running out of fuel. You just have to have some basic monitoring to know the health of your car. And having those lights go on is a good thing. And you don't worry every mile you drive whether you're going to have a light go off. You just operate, and if the light goes off, you deal with it. So I think that's how, so some of this needs to run in the background. Some of the interpretation parts, we need to pull in data in an efficient fashion that we do. I mean, come on, look at Epic, right? Is that the most useless presentation to, you? If you wanted to look at your data for health, and they show you all these charts, it's like some are here, just look at QBios when we show physician how they've organized the data. They go, wow, why aren't we doing this, right? Because they organize it around themes, not by biochemical versus cells versus other things. They operate by immune and these sorts of things that you can interpret. So first of all, it helps the physician a ton to have data organized properly, and it helps you as a person. Let's get it out of the physician's hand to some extent, because you have more time to figure out your health than a physician does by a lot, right? Physician has 15 minutes. That's it. Whereas you have a lot more time, and if something's off, you're gonna follow up. So I'm a big screen on a dashboard. Yeah, that's how it should be. I mean, I'm a believer in the future. The screen's going to be your mirror, right? You're going to wake up in the morning and you're going to get your results back. You'll do pricking and you'll get results back. It's going to show up in your, mirror and say, oh yeah, everything's green. Well, this one's off a little bit. Maybe you better go follow that up. And if it keeps staying up, something's off, right? So I think that's how it's going to roll out. And then you're right about population scale. We're measuring things on individuals to be able to work as across groups of people. We need to find common themes, but I still think you will find a lot at the level of the individual. So I'm a big N of one guy at scale. I think it can be done at scale, actually. And just like everybody has an iPhone, this is going to be your most important health device in the future. More important than your doctor, but it'll work with your doctor. By the way, none of this is going to replace physicians, unintendedly, but it's meant to augment. So I guess that's some of my thoughts on it. I mean, your point, I get your point. It's a valid point. You see where I'm coming from, though, too. Yeah.
Oh my God. And I just want to chime in on that. I had to teach the M1 students, 190 of them this year, human genetics. We are teaching them human genetics from 1975 through 1985. It's so pathetic.
Yeah.
And so I tried to sneak in a little bit of, I actually mentioned your work, Mike, quite prominently, both to our graduate students and to the medical students. And if, you know, this is not going to be on the boards for another 10 years. So we're living in this singularity, and our educational system is about 20 years behind in genetics. So God,
Yeah.
you know, this is a, can you see this?
I can, yeah. Oh, wow. Okay.
Williams is, I mean, this is stuff that's 1955 book that already said this.
Yeah,
And it's
cool. Very cool. Yeah, no, I think, and you know, the medical system is very conservative for obvious reasons. But just think about temperature is a 300-year-old technology to tell if somebody's getting ill from a viral infection, right? It's just, sure, we now have an infrared device on it, but it still doesn't make it any better. Well, why wouldn't you bring in heart rate and heart rate variability as a much, much better measure? Yeah, and then you talk to physicians about smartwatches. Every physician, when I told them about using wearables for a health monitor, oh no, everybody knows they're not accurate. And I said, well, they're much more accurate than what you measure in a physician's office. We've done that, at least for heart rate variability. It's not for temperature, but for heart rate variability, much more accurate, right? I go in a physician's office, I'm up by 40 beats, depending what's going on, by whether I drove in or biked in. Even if I sit around for 15 minutes, it's still up. Or the measurements, you know, everybody assumes because it's in the doctor's office must be right. It's not.
So I have a question on what your thoughts are on for monitoring brain function.
Oh, good question. You know, we're interested, I guess I have two answers to that. One is sleep, we're very, very keen on. And these devices like Fitbit, they're good for sleep duration, not sleep quality and stuff. So there's a device out there called Dream that we're just starting to use now. It's not cheap, it's $1,500. So we're going to try that out. That's supposed to be good. It has all these, you know, sensors on it. That's item one. Item two is I'm a big believer mental health will get monitored by like what we're doing now, by your interaction with electronics, you know, your smartphone, the web searches you do for your mental state. You can tell when people are getting depressed just by their language, their facial expressions. So I think there's a ton to be learned off of, you know, again, visual. And there are companies you may know that are set up around this stuff, MindStrong, things like this. So yeah, Neuralink, somebody put in there. So, and we have integrated some of that. We haven't analyzed the data. We do something called NeuroTrack to look for Alzheimer's in our folks. And we just have the data, but we haven't had time to go through it, which I think will be interesting. So I think that's where a lot of the mental health stuff will work its way in through that sort of format. And the more you can run things in the background without, you know, people really know, without being invasive, the more people do it. Now, some people worry it's too invasive, but you know, there's people out there searching, trying to figure out who's going to commit suicide based on how they're doing their searches and all this sort of stuff. And, you know, I would argue it has privacy issues, but it probably has a lot of value too, right? If you can catch a situation like that and intervene, that would be a positive thing. I think I covered a lot of ground on that. It's a long-winded answer for your question there, but great question.
Yeah, definitely. So I can easily imagine baseline collection, it will be extremely important for this kind of detection.
Yeah, for sure. Train on people's healthy. And along these lines, I'll get in deep water on this, but because I don't mean it this way, but ME/CFS, chronic fatigue syndrome, is something we're trying to do now, measure and make things and get good days and bad days and build training models around this. I don't think that's a mental health, but a lot of people do think that. But anyway, the point is by activity and things like that, we should be able to build these personalized management systems. I would hope for ME/CFS, and I hope for mental health situation, anything with chronic disease, to be perfectly honest, should be approachable with longitudinal tracking. David, I think you're muted there.
So yesterday in Nature, they had a pre-publication of a study with 454,000 human exomes. And I think the goal for that study was to try and find a functional mutation in every human gene. And they said that they didn't achieve that. I think they said it would take 5 million to get to that kind of level. But they got about 15,000. I think they claimed that they found functional mutations in at least one allele. So I just wondered if you want to comment on
You mean disruptive?
huge databases.
Yeah, you mean disruptive mutation, right, David? I would think.
Yeah, they were.
Yeah. But, so unfortunately, I didn't see that. I'm a day behind. Thanks for telling me about that.
It's just brand new. I saw it last night.
Very cool.
It's the Regeneron analysis of UK Biobank, et cetera.
Very cool. Yeah, some genes are kind of hard to find, right? They're super conserved, histones, for example, and
Right.
they're also multiple copies. So some are just going to be playing hard, but yeah, so that's interesting. So you need 5 million people. Yeah, well, okay. But you need those data. We need those data to correlate with, phenotypes to be able to interpret genomes, right? That's really what the bottleneck is in genome sequencing from a health perspective. How do you, right? Oh, somebody put the link in. Fantastic. Yeah. I'm going to click on it myself so I get it up here. So yeah, we need that kind of data to be able to interpret variants. That is a bottleneck. And a lot of the interpretations you may know aren't right. So you need multiple studies to help validate a lot of this stuff. Yeah, thanks for sharing that.
Any more questions?
Please ask questions, because if you don't, I will hog up all the time.
And don't forget, Rob, you do have protected time.
That's why I'm holding back.
So I guess, I actually do have a question. I know, so a lot of the work that you've done have been like bulk transcriptomics, looking at Shannon entropy. So there's a push towards doing more single cell type omics in the brain and the body. So how do you envision that fitting in? Is this going to be tremendously useful, or is it, what do you think?
So I'm sure the answer to that is yes, right? If we can see cell states, we just don't know what to look for. Right now, it's prohibitively expensive. And we haven't done, we're going to do a whole genome-based sulfite sequencing on our whole set of samples. So it's just so expensive that we've held off. I didn't want to do a raise yet, which I know is very doable for these samples, but even though we collect a lot of material, it's getting very precious now. So we're trying to be very judicious. And so I think those will both be powerful, but single cell, I think, will be powerful, and especially in certain areas like the blood diseases, to know whether they're clonal or not. You can do some of that already just by, VDJ things, but I think we can learn a lot more. So the answer is, it just doesn't format well right now. It's very expensive. It's very slow. And maybe in the future it will, as these prices drop, it will make sense to bring that in. And so we keep our eye on it all the time. And then people say, well, how would you use spatial information? Obviously, we're in blood, so spatial is not so critical right now. We're very intrigued to bring in, you may know the GRAIL stuff. Well, this group of all people would probably follow the GRAIL work, where you can follow 50 cancers. A lot of it's late. I think that's a little oversold. A lot of it's late stage stuff. That's still better than nothing, I suppose. So we want to incorporate those kinds of tests. I think that would be very interesting. It's not single cell, but there's lots more to do, that's for sure. And we're always evaluating.
So, I have a question. You alluded to it a little bit. So, if I had to say a theme is, you want to collect information that is easy to collect. Heart rate is easy, maybe blood pressure, glucose monitoring. The other thing that comes to mind is stool and urine. Those are also relatively easy. Blood is harder. You have to poke and stuff. I'm sure you have done this. So can you tell us a little bit more about what you found in terms of urine and stool?
Yeah, so we did the microbiome, as you saw from the stool. And we've done some metaproteomics and metametabolomics from stools on our list, just haven't gotten to it. So we're very interested in the microbiome, right? So is everybody. Everybody knows the microbiome is really, really important for your health, yet nobody's really figured out how to incorporate it into a clinical measurement that people can act on. So people have used it for discovery, and now, Second Genome has a new name, but they're doing, they have a probiotic out from that work that they think helps for diabetes. And data look pretty good, actually. So I guess we're still in the discovery mode. Yeah, did you say Solnock? Yeah. So anyway, we're in the discovery mode for that. Urine, I didn't say, but we are doing some profiling there. We're doing a ton in pregnancy to try and follow the gestational age, preterm, things like that. So I guess just the way the project's rolled out, it should do more with the IPOP study, but we've done a lot with pregnancy because, that's a group that wants to get followed pretty closely and for both all those things, preterm, delivery date, that sort of thing. So that's kind of where we are. And we have some nice markers, I can say, on the pregnancy things. I think one of them's up on bioRxiv. It's about to get accepted, I hope. You're hitting spot on. I think those things could easily turn into simple analyte tests, much like, just knowing if you're pregnant, right? You can, that's where we need to get to on this stuff. But I'd like to see the multi-analyte where you pee in something. You've seen the smart toilet stuff probably too. You pee in something and 6 analytes, or in the future, maybe 50 analytes get measured and give you some readout for a certain number of health conditions. That's would be nice. And if it's automatic, right, you pee in your toilet and get out the results, it's even better. So be terrific.
I have a related question to all this blood analysis. What do you think about, some people are not doing this, taking PBMCs and then challenging them with different agents? So, I mean, you're not looking at the status quo, but sort of the range of reactivity and the directions of reactivity of an individual, which is much under certain biological stress.
I think that's a biggie. We're doing some of that too. Yeah, so I think that's really, it is a good thing to do. It doesn't format that well into what we're doing. What I didn't talk about is we often save up several years of samples and then analyze the data all at once, avoid batch effect. There's a certain number of things that are all done, real-time absolute values. That's expensive to do. So we've been doing, because we're doing omics technologies, and especially the mass spec stuff is very, very prone to batch effects. So we've saved up, so that makes it trickier. And so the PBMCs would be, you'd have to do them kind of one-off. They work best fresh, although you can do them frozen. It's true with some of these LPS things. Yeah, you're shaking your head because it's not quite as good. Anyway, I think there's value. We were trying to figure out some immune issues, and we did see that the stimulated assay was much more informative than the non-stimulated one. So you're hitting a really great point, I think, and would be worth bringing in at some point. Yeah, lots to do.
Keep going, folks. No dead air. No dead air.
Lots of great questions already, that's for sure.
Megan, I'm delighted to open up my time with Mike to anybody that wants to join us. If you want to share it with the whole team, I can give Mike and anybody who wants to join a sort of a prelude of where this is likely to go. How can we make animal models relevant to accurate human healthcare? Because there's a huge chasm there. In the same way that EHR data is crap, basically, most of the mouse and rat data that we generate is completely irrelevant, painfully so, because it's all based on C57 Black 6. And we're in an enviable position here in Memphis to be pushing true, accurate healthcare for mice and rats. And I just want to see whether Mike can give us some advice on how to conciliate. And there've been a sort of a thread in the background from Klaus and others, like, wouldn't it be cool if we could get Apple Watches for mice? Because the things we could do then would be pretty fantastic, because we can replicate the genome as many times as we want in as many environments as we want and actually get ROC functions for outcomes. But anyway, so. But
Do you want me to
I'm gonna
comment
shut
on
up.
that now, or we're going to save that for our 101 there? Or,
I mean,
Should
please
I-
go ahead unless,
Do you want me to stop the recording now or?
no, this is going to be good. Let's keep it going.
well, I
I'll
do
just have
worry. Yeah, I mean, I
Where?
do worry, same as sounds like where you're going with your comment, that mice, they're great for many things, great for basic developmental processing things, but as models for human disease, you've heard it said many times, we've cured cancer many times over in mice, where we haven't done that in humans. And there's just a big enough difference, especially in the area of diabetes, right? The metabolic profile of a mouse, 600 beats per minute heart rate, is so different from a human that the modeling, again, there's some conservative stuff. So don't get me wrong, when you can learn things, it's wonderful. But I don't think it is the best. We've stayed away from mice, as you probably know.
I don't blame you.
Yeah, just
would
we
say
find
that-
mice too hard. But on the wearable stuff, that's interesting. We have a project going on because we want to see if we can pick up cancer from physiological data, like a smartwatch. And we have our devices on people, and they are getting heart attacks and things are pouring over the data. It's hard to get it's not a controlled system very well. So we're trying to do some things with mice with that. We'll see how it works. All I can say is stay tuned. We're just getting started to look at all the same things you'd find in a smartwatch. Look at it on a mouse.
Yeah.
With
I just want to argue about one thing. So when people tend to say it doesn't work in mouse, they mean it doesn't work in one particular haplome of mouse. It's not even a real mouse. They mean C57 black 6, fully homozygous mouse. And we just spent $100 million studying this particular mouse, and it doesn't look like humanity. Isn't that strange? And it would be like taking Mike Snyder and saying, oh, isn't it funny that Rob Williams and Megan Mulligan don't look just like Mike Snyder? And you made the point exactly yourself, that one human is more different. The disease state changes are less than the differences between normal humans. That's what mouse genetics, rat genetics has suffered for this reductionist mindset from the beginning of time, basically. And it's tragically misdirected.
Fair point, Rob.
And-
Yeah, and even more so, right? Because in nature, mice variation is much bigger, certainly at genetic level, than humans, which went through a bottleneck. So yeah, your point's really well made. And not only that, they're in a very controlled environment. I think we tease things out. I talk about picking up infectious disease from resting heart rate. My own prediction is that we do better. We get a walking heart rate, kind of like a stress echo, right? You actually tease out defects. You see them better when you stress people a little bit. So I think if we have mice living, maybe they're stressed because they're colder than they're supposed to be, but they're still in a cage, living the fine life, sort of, I guess, as opposed to out in the real world doing whatever mice do in the real world. Watch out for cats and all that sort of stuff. So your point's well taken.
So David Ashbrook is on, I think, still. Yeah. So we've been doing some G by E studies with David and Shuhida and Shaunak and others that are really pretty cool with the mice, where and we can actually do the indirect genetic effects also with the mice, which are also cool. So I guess at some point I would love to show you some of the results we've gotten in recent studies where we've done aging in mice. By mice, I mean 40 to 80 different genomes of mice, longitudinal studies. We then can sacrifice them at different time points, isogenic cohorts. So now we have metabolomics, proteomics, transcriptomics, et cetera, from the same quote individual with quotes around it, over the lifespan. And we know what the mean lifespan of that mouse is in condition A, condition B, potentially condition C through Z. And oh my God, they're all over the place. Some mice live longer on a high fat diet, like 60% fat. Some mice on that 60% fat don't gain weight. Others will crump a year earlier than other mice. So it's basically recapitulating the points that you make. But unfortunately, the rodent model genetics, which is really not genetics community, it's really all molecular biology, functional genomics. They don't get it. They don't want to increase their workload.
Yeah, they claim, well, it's not controlled, but that's the point. It's how the real world is. Yeah, the real world is not controlled like that. So yeah, I think we would pretty much agree on that.
Yeah, just jump in on top of that. Benny, one of our collaborators, has done the methylome aging, and she has a really similar plot to what you showed of chronicle age versus pharmacological age, and the bunching of particular strains far more than between strains. So there's clear genetic control of that sort of chronological age versus phenotypic age.
Very cool. Yeah, I would say, in every perturbation we've done, we have people doing weight gain, weight loss. I didn't show, but a fiber study where people with fiber, the VO2 max exercise study I mentioned, viral infections, they always cluster by person, every single one. The one where they're not clustering as much by person is in cancer, where we've looked at the samples might be clustering a little bit more by, so that might, there the perturbation may be much stronger and enough to create it. But that's so far, that's the only case, even to some extent when you're early cancer, we're looking at familial adenomatosis polyposis and looking at polyps. So even some of the early ones, right, they'll cluster by person, not by whether you're early stage cancer or not. So yeah, I just think people don't appreciate that. And that's why they don't get them too. And even in today's world, right? I'm a good example. If you look at your clinical mark, we all probably have this, right? I'll be marching along low for certain markers. I'm still in the normal range, but suddenly it'll jump up. It'll go up twofold on my next visit. It's still in the normal range, but it's just below the bar. And any physician generally looks at the static measure and says, all right, it's fine. You're still in normal range, but that trajectory is not a good one. And this happened to a friend of mine who was in the IPOP study. He said, Mike, my ALK went way up. I'm still normal. I said, that's interesting. He took his next one because he saw this one was high, and sure enough, it was even higher. So he clearly was heading down an adverse trajectory. And so he changed his diet. Actually, it was a pretty strong liver marker, and it cleared it right up. But it's only because he was kind of on top of the trajectory that he caught this, and not so much on the absolute values. Yeah, so yeah, it sounds like your aging stuff's pretty cool. I'd love to see some of that. Well, maybe we can look at that in the next few minutes, Roy, or Rob, if you want.
Yeah. Yeah, David's mixing in the Alzheimer's disease human gene. So we're taking human transgenes that confer high susceptibility to the familial forms of Alzheimer's, and then asking to what extent we can modulate AD risk in mouse, and maybe even eventually get to the prevention stage with these particular human transgenes banging around. But for example, just coming back to your original comment, so the Alzheimer's disease models of mice have failed because they were all done on a black 6 background. If you mix in a lot of variation, you see almost precisely the same wide range of disease onset with the presenilin 1, with the A beta, with the ApoE. So you'll get a 20-year range in mouse equivalent terms.
Wow. And these are all different wild type strains, Rob? Is that what's going on here?
These are all, it's the BXD family, so they're segregating for about 6 million variants.
Okay.
It's just a family, but it's still enough to shake things up.
Got it. Cool. Yeah, people are.
Anybody else?
I think people are intimidated by these complex systems, and I personally like them because I see them as a challenge to take on. And plus, you bring on these things. Yeah, if it's simple, why we just study it? We'll figure it out. Let's do the things you can't that are harder. But these days, with the computational tools and things that are out there, you can start bringing in lots of data and make, again, especially the machine learning stuff. I didn't show you But take a look at our preprints on this ALS and the COVID-19 severity. It's pretty sophisticated, just this brilliant postdoc I have. You may want to hire him actually. He's a brilliant, and he just takes these things. In fact, one reviewer, we are having trouble getting published. One reviewer said, "Well, there can't be that many genes." That's the only way. We show it like 85 times why this is all significant. It doesn't matter. You just can't believe it. So the tools are there to unravel complexity at a level that's never been possible. So. Yeah.
So, Mike, you're one of the few people that's been in human genetics for a long time. You gave a talk that didn't really even mention GWAS. You used the polygenic risk scores at the front of your talk. There's sort of a counter current because on the one hand, you're kind of saying, okay, polygenic risk scores aren't a total waste, but given all the environmental complexity and the fact that they're not longitudinal, yeah, maybe they are kind of a waste. So where do you come down on this mania now for, oh, we're going to compute your polygenic risk score and tell you what to inject when, where?
I think, all right, I'm about to offend some folks. I think polygenic risk score is on its way out. It does have some value now. And why is it on its way out? Because it's this problem I said before, it's linear. Genetics is just not linear. And it doesn't bring in other features as well. I think it's just, again, yeah. So I won't say more than that. But I think this is where machine learning kicks in. And so it works. Again, if you're like, it worked for me in diabetes, so I can't possibly rule it out. I first predicted, I didn't, told Butte that he did my polygenic risk score and said, Mike, you're at the way high end. Shows the distribution of everyone. I'm like way out there. And son of a gun, I got diabetes after a viral infection. Classic genes plus environment, most likely, right? So anyway, yeah. So I just think we can model all that stuff, right? All these measurements, they're features, whether it's a genome sequence, whether it's EHR measurement, whether it's what have you. These are all features that can be fed into equations. So to figure out how to better weigh them and that sort of thing. And then we can learn from all of them. Let's use all the information and not assume, yeah, things that probably aren't right anyway. And of course, again, GWAS is missing all the rare variants, right? And we all have like, was it 4 million rare variants? Or maybe it's 2 million. Depends on where you put the cutoff, I suppose. We have a ton of rare variants. Everyone. And there's a whole bunch inside your gene itself, not just in the external, and none of that's modeled in. And so take a look at our COVID severity paper. Again, it's not where I want to be yet, but it's pretty darn close to where I want to see us get with genome analysis. So it's a step in the right direction. Yeah. That's the right way to say.
Yeah, no, those are great comments. The one thing, I've been, when I had to teach the medical students, I had to relearn quite a bit of genetics. So I went back to the missing heritability paper from 2009 or 2010 that everybody and their mother signed on to and read all those comments. And it was sort of I just slapped my head because it seemed like there was so much obvious misunderstanding, because all of these, the missing heritability is a concept that really is appropriate if you're looking at selection coefficients and evolution of a species where the effects are additive. The only, that Mother Nature selects for additive effects. She can't select for epistasis very effectively because it's unlinked. It's all over the place. But if you're trying to make a prediction for an individual, I'm just loaded with epistatic interactions. So there's this difference between the population geneticists on the one side that kind of live in their own little bubble, and then the people that should be trying to, what you're doing, give me longitudinal data for me or you, and let's see what we can predict about me, and let's see what we can put together in terms of epistatic interactions and nonlinearities for me that's going to be different from everybody else to come up with an ROC function. So
yeah, my comment it's worth some of that would be through your complex genetics of you, but some of it's because of the context in which you live, your environment. So I think that's what you're getting at, right, Rob? That you're all, so these things show up because you're in a certain environmental perturbation. Environmental context is probably a better terminology there. That's why. So again, genes and environment. But I'll tell you, with the machine learning, we took I think the heritability for ALS was something like, was it 17%, maybe a little less. Don't quote me on these numbers for sure. We found just by the machine learning, which does bring in other features, you know, we're at 77%. We capture a hell of a lot more. Again, that freaked out this reviewer who just doesn't like these numbers, who just says, "Ain't it?"
So you're painfully familiar with the overfitting critique.
Yeah, but we you know, deal with all that, right? It's machine learning first of all, so you can, yeah, but you have to watch out for machine learning there. But then we can validate this stuff by lots of means. And these days we'll often take our common analysis with the GWAS, where we machine learn with GWAS. We can validate a lot of it with the rare variant because a lot of the genes turn out to be the same. Not entirely, of course, for obvious reasons, but you can get some cross validation from the two different approaches. Kind of cool.
David, why don't you summarize the sequencing effort that you're doing for the BXD family? I think that'll intrigue Mike.
Yeah, if you want to show slides, I'd love to see them too, whatever works. Yeah.
Yeah, I can talk while I try and look for slides at the same time. So
if you don't mind, I can use the restroom 1 second.
That'll work.
I'll be right back. That's the one minus not getting that break there.
What a great talk that was.
Yes.
Hey, Lulu. You guys can all unmute if you want. At this point, we can be as noisy as we want.
Sure, I'm here.
My wife has a good Lyme disease story too she could sell. But when Mike comes back, maybe.
David, will have more talk?
I was just going to talk to Mike and just sort of explain the BXD family to him a bit, maybe a little bit about DO, diversity outbred, and a bit about the sequencing. Rob just wanted me to.
Sure. I'm going to go ahead and stop the recording now because it's going to get long, unless you want me to keep you on. Okay.
No. Go ahead and cut it if you want.
Okay. I just realized, well, Arthur can do some editing.
I mean, if you hang in there, Megan, you could do a second recording that we could you could send it to David and whoever else is interested.